Today I will talk about skeletal tissues, which are subdivision of the connective tissues of human organism. And namely, I will talk about cartilage and bone, which are forming skeleton. Cartilage is a tough, resilient type of connective tissue that structurally supports sedents of tissues, notably in the respiratory tract, and provides cushion low friction surface in joints. Cells of cartilage called chondroblasts and chondrocytes make up a small percentage of the tissue's mass, which is mainly a flexible mass of the extracellular matrix. Chondrocytes are embedded within lacuna, surrounded by extracellular matrix. Cartilage extracellular matrix typically includes collagen as well as abundant proteoglycans, notably aggrecan, which bind a large amount of water. Cartilage always lacks blood vessels, lymphatics, and nerves, but it is usually surrounded by a dense connective tissue perichondrium that is vascularized. There are three major forms of cartilage: hyaline, elastic, and fibrocartilage. Structure of the cartilage. It consists of the perichondrium, which is enveloping the cartilage itself, including outer fibrous layer consisting of collagen fibers and fibroblasts, and inner cellular layer consisting of chondrogenic cells and chondroblasts. Chondrocytes forming isogenic groups of cells located in lacunas with integrin ensuring glycoprotein in their plasma membrane. Chondroclasts are in charge of cartilage resorption and remodeling. Cartilage matrix includes type 2 collagen fibers and elastic fibers, especially in elastic cartilage. Ground substance consisting of aggrecan, proteoglycan rich in hyaluronic acid and chondroitin sulfate and carotene sulfate glycosaminoglycans, and chondronectin anchoring glycoprotein of cartilage tissue. Here you have the diagram of the typical cartilaginous tissue, including the fibroblasts in perichondrium, chondroblasts and chondrocytes in the cartilage itself. It's in the extracellular matrix. There are the recognized interterritorial matrix in between the lacunas covered with chondrocytes, and territorial matrix, they immediately surrounding the chondrocytes. Here you have the isogenic groups of cells. Here is the isogenic group of cells, two cells, here two cells, here two cells in one single lacuna. This is perichondrium, this is superficial chondroblastic layer and deep layer of chondrocytes and extracellular matrix. Chondroblasts are immature cells located in the periphery of the cartilage, actively secreting extracellular matrix, which consists of type 2 collagen and or elastin, as well as proteoglycan micromolecules, aggregans namely. Chondrocytes are forming isogenous cells aggregates located in the lacuna, surrounded by a large amount of territorial and interterritorial matrix. Proteoglycan aggregates attract a huge amount of water molecules. Due to this binding impact of water in cartilage consists about 60–70% of total tissue weight, but it is not water which can be removed from the cartilage. It is water which is connected with the glycosaminoglycans with the complexes of proteoglycan micromolecules. Here you have the typical chondrocyte located in the lacuna. Surrounding is the extracellular matrix. In the chondrocyte, you see the nucleus, nucleolus, the rough endoplasmic reticulum, the Golgi complex, and the vesicles which are containing the elements of extracellular matrix of chondrocytes. Here we have the isogenic group of chondrocytes surrounded with the ground substance and fibrous elements. Rough endoplasmic reticulum present Golgi complex, euchromatinized nuclei, and mitochondria, which are necessary for the energetic supply. Extracellular matrix of cartilaginous tissue. Here you have the collagen, type 2 collagen 5 fibril, which is having cross striations, including the dark and light bands. In between the collagen fibers, there are present proteoglycan micromolecules, which include hyaluronic acid, connected to the by linking protein with the core protein, and to this core protein, chondroitin sulfide and dermatase sulfate and other sulfated glycosaminoglycans are attached, and they bind a huge amount of water molecules. That is why the cartilaginous tissue is highly hydrated. 60% of the total weight of the tissue is represented by water element. Location of different types of cartilage. Hyaline cartilage is found as the structural framework for trachea and bronchi, on the anterior ends of the ribs, on the surfaces of synovial joints, constitutes much of the fetal skeleton. Elastic cartilage is found in the auricula of the external ear, in the auditory tube, in epiglottis, and in part of the larynx. Fibrocartilage is found in the intervertebral discs, pubic symphysis, and in some joints. Here have the location of the hyaline cartilage, which is depicted with the blue coloration, interfacing the interface between the ribs and the sternum, epiphyseal parts of the long bones, auricular, articular cartilage of the joints. The fibrocartilage is present in vertebral discs, in symphysis pubicum, and elastic cartilage is located within the external ear, auricula, and also in epiglottis, and that's it. Extracellular matrix of hyaline cartilage is homogeneous and glassy, rich in fibrils, type 2 collagen, and aggregate complexes with bound water. The extracellular matrix has less collagen and more proteoglycan immediately around the lacuna, producing slight staining differences in the territorial matrix. Chondrocytes occur singly or in small, methodically derived isogenous groups of cells. Perichondrium is usually present, but not at the hyaline cartilage of the articular surfaces or the epiphysis of the growing long bones, as well as in the fibrocartilage. Here you have the light micrographic representation of the hyaline cartilage, showing you perichondrium, chondroblasts on the periphery of the cartilaginous tissue, isogenous groups of chondrocytes located in the lacuna in the cartilage itself, and surrounding dense connective tissue in the perichondrium and adipose cells, which are present everywhere in the organism, filling the gaps between different elements of the connective tissue. Here you have another high magnification of the perichondrium and cartilage isogenic groups of cells located in lacunas. Territorial matrix is surrounding chondrocytes, and the interterritorial matrix is located between the isogenic groups of chondrocytes. Here you have the two isogenous groups of sonochondrocytes located in lacunas with lipid inclusions, with nucleus present, with synthetic machinery represented by rough endoplasmic reticulum, Golgi complex, and mitochondria in the cytoplasm of the chondrocytes, which are secreting extracellular matrix. Elastic and fibrous cartilage. Elastic cartilage generally resembles hyaline cartilage in its chondrocytes and major extracellular matrix components, but its matrix includes abundant elastic fibers visible with special stains, which increase the tissue's flexibility. Elastic cartilage provides flexible support for the external ear as well as certain structures of the middle ear and larynx. It is always surrounded by perichondrium. Fibrocartilage contains myelinic combinations of hyaline cartilage in the small amount of the dense connective tissue. Histologically, fibrocartilage consists of small chondrocytes in the hyaline matrix, usually layered with larger areas of bundled type 1 collagen with scattered fibroblasts. Fibrocartilage provides very tough, strong support at tendon insertion and in intervertebral discs and certain other joints. There is no distinct perichondrium in fibrocartilage. Here's the elastic cartilage with arrows showing you the isogenous groups, elastic fibers, and ground substance covering the space between the isogenous groups. Fibrous cartilage of intervertebral disc, including chondrocytes located in regular manner in parallel arrangement, consisting of the type 1 collagen fibers. And this is the annulus fibrosum, the ring covering the nucleus pulposum of the intervertebral disc. And here you have the element of the bone tissue, which is forming the vertebral tissue. Here you have another view on the light micrographic representation of the fibrocartilage with chondrocytes forming the parallelly arranged rows of the cellular elements. In between, the collagen fibers are forming of collagen type 1. Collagen fibers of type 1 collagen are being located. Here you have an increased magnification there. Chondrocytes, collagen fibers, bundles, and this is the general arrangement of the fibrocartilage. All forms of cartilage form from embryonic mesenchyma. So it is one single origin for the whole differences of the connective tissue. Cartilaginous structures grow by mitosis of existing chondroblasts in lacuna, interstitial growth, or by formation of the new chondroblasts peripherally from progenitor cells in the perichondrium. It's called appositional growth. Repair or replacement of injured cartilage is very slow and ineffective, due in part to the tissue's avascularity and low metabolic rate of chondroblasts. So, in order to repeat the most important things, that cartilage is highly containing a large amount of water molecules bounding to glycosaminoglycans, is hard tissue, the cellular components, including chondroblasts and chondrocytes, avascularity, predominance of the extracellular matrix, and depending on this, consists of the in extracellular matrix, it depends the classification of the cartilage tissue to the hyaline, elastic, and fibrous cartilage. The next item for today's presentation is the bone tissue or the osseous tissue. You have the bone fragment of the long bone, the epiphyseal part of the long bone segment, showing you components of the osseous tissue, the bony lamella, the aversion system, trabecules, compact bone, spongy bone, outer circumferential lamella, and periosteum, which is including the blood vessels and the poorly differentiated cells giving birth to the regeneratory and replacement of the damaged bone tissue. Bone is a type of connective tissue which, with a calcified extracellular matrix specialized to support the body, protect many internal organs, and act as the body's calcium ions reservoir. Osteoblasts differentiate from stem osteoprogenitor cells and secrete components of the initial matrix of the bone tissue called osteoid that allow matrix mineralization to occur. Important components of osteoid include type 1 collagen, protein osteocalcin, which binds calcium ions and matrix vesicles with enzymes generating phosphor ions, PO4 ions. High concentration of calcium and PO4 ions cause formation of hydroxyapatite crystals whose growth gradually calcifies the entire matrix. Here on the picture you have developing osteoblasts and osteocytes in the developing osseous tissue. These osteoblasts are inactive, they are sleeping, they're hibernating, and osteocytes located in the lacuna are visible, surrounded with the calcified matrix of extracellular matrix of the bone tissue. Here you have the osteoblasts, which are changing from the flattened squamous-like cells to the columnar or cuboidal cells involved in the active secretion of the extracellular matrix. And this extracellular matrix is having coloration of the dark pink one. Here you have mesenchyma or magenta red extracellular matrix. Here you have electron micrographic representation. It's a talk in previous lecture that clear recognition and destination, the definition of the connective tissue cells is available with the electron microscopic magnification, which opens the details of the morphology of the structure and of nucleus and of cytoplasm of the cells of connective tissue. The same is in the osseous tissue, osteoprogenitor cells located on the top of the osteoblasts. Osteoblasts are located on the periphery of the secreted extracellular matrix material. We have 5 osteoblasts which are cuboidal in form. We have cuboidal shape, which is encompassing that they are actively secreting the extracellular material. And here you have the osteocyte, which is incorporated in the lacuna, in the osseous lacuna, surrounded with watery fluid. And processes of the osteocytes are directed toward the osseous or osteoid extracellular matrix, communicating with the neighboring osteocytes, forming the functional syncytium of the whole osseous tissue. Here is the mechanism of bone mineralization. This is the layer of the osteoblasts, which contain the secretory vesicles accumulated in the upper portion of the osteoblast and moved via the exocytosis to the extracellular space. Secretory vesicles, after emerging from the osteoblastic space, are colored with the yellow coloration. According to the activity of the alkaline phosphatase and pyrophosphatase, the phosphate ions are being formed in the extracellular space surrounding osteoblasts. This is called osteoid layer. The accumulation of the calcium phosphate cells or calcium hydroxyapatite crystals is advancing and developing. Secretory vesicles showing early mineralization and secretory vesicles in near confluent mineralization, and then mineralized bone, which is consisting of hard tissue, hard extracellular matrix, due to the 70% of calcium hydroxyapatite crystals present in the bone tissue. In cartilage, 60–70% of the cartilaginous tissue is presented by the water molecules, and in the bone tissue, in osseous tissue, about 70%, 72% are represented by the calcium hydroxyapatite crystals, which are making hardened, confluent mineralization area. Osteocytes differentiate further from the osteoblasts when they become enclosed within matrix lacuna and act to maintain the matrix and detect mechanical stresses on bone. Osteocytes maintain communication with adjacent cells via the network of long ramifying processes that extend through the matrix via narrow canaliculi radiating from each lacuna. Osteoclasts are very large cells formed by fusion of the several blood monocytes or stem cells of the red bone marrow, which locally erode bone matrix during osteogenesis and bone remodeling. Here you have the high magnification light micrographic representation of the osteocyte embedded in the lacunas, forwarding their processes in the bony canaliculi in order to contact the neighboring cells and form the functional syncytium of the whole living tissue of the bone tissue. Here you have the osteocyte in lacuna on the electron microscopic level. The cell machinery of the synthetic activity is present. Golgi complex of endoplasmic reticulum, secretory vesicles containing microvesicles with calcium ions. Exocytosed move to the osteoid and binding to the phosphate ions, forming hydroxyapatite molecules or crystals, which are hardening the extracellular matrix of the bone tissue. Here the both light micrographic representation of the osteocytes, the osteoclast, osteocyte, lacuna space filled with watery fluid, cell process of osteocytes present in the or directed toward the canaliculi of the bone tissue. This cell is typical spindle shaped with the nucleus containing much of the eochromatin involved in the process of the synthetic activity of, besides the calcium, mineral calcium vesicles. It consists abundance of the proteins, the osteocalcin, osteogenin, osteoprotegerin, and other enzymes which are involved in the process of the mineralization. Here you have the osteoclast in the H. This is house ship's lacuna. in the lacuna, which is the site where the resorption of the bone tissue takes place. Each osteoclast consists several nuclei. It is polyploid cell, and it secretes the carbonic acid and secretes catalase, which is metallic proteinase, resorbing both hardening calcium phosphate hydroxyapatite crystals by the hydrochloric acid, which is formed from the carbonic acid in the house-shaped lacuna, and then resorbing the collagen fibers by the activity of catepsin molecules, catepsin enzymes. Here has increased its biggest available magnification of the osteoclast located in house-shaped lacuna. You have several nuclei with prominent nucleoli. Here you have another osteoclast. Here you have the osteoblasts, which are building the new bone tissue instead of the tissue which was resorbed by the osteoclast. And this resorption and regeneration is the constant physiological process which is taking place during the whole lifespan of the organism. It's long-lasting remodeling and regeneratory process in the osseous tissue. Here you have the electron micrographic representation of the osteoclast present in the house-shaped lacuna. These are the ruffled border from which the carbonic acid is delivered to the zone of resorption. And here is the sealing contacts, junctions, which are sealing the house-shaped lacuna, preventing the resorption of the bone in other place than the house-shaped lacuna. Osteoclast on the light microscopic magnification, high magnification of light microscopy, 1, 2, 3, 4, 5, 6 nuclei. Here you have another osteoclast, lacuna of house-shaped and bone tissue, which is subjected to resorption. Osteoclast structure and function. Several nuclei, abundant mitochondria, Golgi complex, which is secreting lysosomes delivered to the house-shaped lacuna. This is the ruffled border from which the, due to endocytic vesicles, carbon dioxide and water molecules are forming carbonic acid, and further carbonic acid is subdivided to protons connecting with the surface of the bone tissue is responsible for the resorption of the calcium hydroxyapatite crystal molecules, and the catepsin is resorbing the organic component of the bone tissue, namely collagen fibers. This is the circumferential clear zone, which is sealing the site of the resorption, sealing the house-shaped lacuna from the dissociation of the process of resorption in other sites except the house-shaped lacuna. Osteoprogenitor cells. This is a summary of osseous components, cellular components of the osseous tissue. Osteoprogenitor cells listing cells that can transform into osteoblasts. Osteoblasts are interdifferentiated bone-forming cells that secrete bone matrix. Osteoid. Osteocytes, the major bone cells residing in lacunas enclosed by bone matrix, previously secreted by osteoblasts. Osteoclasts are large multinucleated cells whose function is bone resorption. Extracellular bone matrix of the tissue. Non-organic component of bone matrix represented mainly by calcium phosphate salts, calcium hydroxyapatite, namely crystals which with formula calcium 10 PO4 times 6 times and OH twice, covers up to 65-70% of total bone weight. The rest, 30-35% of the bone matrix represented by organic component called osteoid consists mainly of type 1 collagen fibers as well as proteoglycan complexes like aggregans. Osteoid glycoproteins, osteonectin, osteocalcin, osteoprotegerin, bone sialoprotein are responsible for binding of cellular components with extracellular matrix constituents. Osteoclasts bind calcium ions to matrix vesicles, while enzyme alkaline phosphatase and pyrophosphatase generate PO4 ions. High concentration of calcium of both calcium ions and PO4 ions cause formation of hydroxyapatite crystals, whose growth gradually calcifies the entire matrix. Osseous tissue and bones relationship to osseous tissue and the bone. Bone is a type of connective tissue characterized by a highly mineralized extracellular matrix, 65% of calcium hydroxyapatite crystals and 35% of type 1 collagen fibers, and aggregant proteoglycan complexes. Non-calcified extracellular matrix is called osteoid. Osseous tissue is classified as follows: primary or woven bone, developing directly from the mesenchyma. Secondary or lamellar bone includes subdivision into compact, dense bone and spongy cancellous bone. Bones are covered by periosteum, a sheath of dense connective tissue. Bone cavities are lined by endosteum, a delicate layer of connective tissue, cells and fibers deep inside the bone. So bone is the organ. Osseous tissue is the bony tissue which is forming the organs, either flat or long bones of the skeleton. Dense bone immediately beneath the periosteum is called compact bone. Deep to the compact bone are small bony trabecules or spicules of cancellous or spongy bone. In long bones of the limbs, there are two types of major bone tissue occur in both the knobby bulbous ends called epiphyses and intervening shaft of the diaphyses. Immature bone called woven bone is formed during intramembranous osteogenesis or repair and has a calcified matrix with randomly arranged collagen fibers. Structure of adult long bone. Gross anatomy including diaphyses, two epiphyses, and two metaphyses, or metaphyseal plates. Units of diaphyseal bone structures are called osteons. Each osteon includes concentric lamellae, collagen fibers arranged in parallel manner. So lamellar bone is consisting of collagen fibers which have parallel orientation in the space. Eversion canals with blood vessels, osteoblasts, and osteoprogenitor cell inside. And osteocytes located in lacunas forwarding their process into the bony canaliculi in between the lamellae. Besides osteon, diaphyses consist of interstitial lamella, circumferential lamella, perforating Volkmann canal, periosteum, and endosteum. Here you have the small magnification representation of the compact bone and spongy bone or cancellous bone, consisting of bony spicules and bony trabecules, forming the framework, forming the supporting elements for the developing bone for the functioning bone marrow in the spongy bone of flat and of the long bones. Bone mammalian Haversian system. By the action of osteoblasts and osteoclasts, woven bone undergoes rapid turnover and is remolded into lamellar bone with new matrix deposited in distinct layers with parallel arrangement called fibers, bundle called the lamella. Both compact and cancelon bones is lamellar bone. Most lamellar bone consists of lamellae organizing concentrically around small central canals containing blood vessels and nerves. This organization is called an osteon or Haversian system. Within each osteon, osteocytic lacuna occur between the lamella, which canalically radiating through the lamella, which allow all the cells to communicate with the central canal, forming the functional syncytium. Periosteum is a layer of dense connective tissue on the outer surface of bone, bound to bone matrix with bundles of type 1 collagen called perforating or Sharpey's fibers. Regions of periosteum adjacent to bone are rich in osteoprogenitor cells and osteoblasts that mediate increases in bone thickness by appositional growth. The endosteum is a thin layer of active and inactive osteoblasts which lines all the internal surfaces within the bone. Osteoblasts here are also required for bone growth. Here you have the histological view on the long bone structure. Epiphysis, epiphysis, metaphysis, metaphysis, diaphysis. Segment of diaphysis showing you the outer circumferential lamella, periosteum covering it, the osteons of the Haversian systems, the cylindrical shaped arrangement of the bony lamella and osteocytes, which are hiding inside blood vessels, the osteoblasts and osteoprogenitor cells. Here you have the increased magnification of the osteon, the direction of the collagen fibers within each lamella is on the cross, on the right angle to the neighboring lamella, and osteocytes located in the lacunas are present in the wall of the osteon, in the wall of the Haversian system, forwarding its process to the bony canaliculi. Here you have the spongy bone consisting of trabecules. Each trabecule is covered with osteum, having osteoclasts, parallelly arranged bony lamella and osteocytes located in the lacunas of the osseous tissue. Epiphysis, diaphysis, medullary cavity, periosteum, compact cortical bone, medullary trabecular bone. And diagram of the epiphyseal part of the long bone showing you the osteons, 3 osteons with central located canals within each the blood vessels are traveling through the bone tissue. The Volkmann canals perforate located on the right angle to the Haversian canals. The outer circumferential lamella, the periosteum, the spongy bone, trabecular bone, both osteons and trabecules are consisting of the lamellar bone. And that's it. Another view of the osteogenic, osteo-Haversian systems. Inner circumferential lamellar, outer circumferential lamellar, periosteum, Haversian canal, lacuna, endosteum, medullary trabecular bone. interstitial systems, the bony lamella covering the space between the cylindrically shaped Haversian systems of the osteons. And here you have the Volkmann canal perforating in the right angle with respect to the Haversian canal, right angle from the surface periosteum. Here the light micrographic representation of the epithelial part of the long bone, showing you osteon with Haversian canal and with the osteon lamella, with cells, osteocytes present in the interstitial space and inside the lamella. These are osteocytes located in lacunas. Here you have the osteocytes, both osteocytes located in lacunas, and here you have the periosteum composed of the outer circumferential lamella surrounding the osteons present in the inner part of the diaphysis. Here you have the several osteons with Haversian canals, interstitial lamella, bony Haversian system lamella, and perforating canals of Volkmann connecting the Haversian vasculature to the periosteum vasculature, supporting with the blood and nutrition, oxygen and nutrition support giving. Osteocytes with processes forwarded to the bony canaliculi, three parallelly arranged, neighboring arranged three lamella, Haversian canal. And this is one single osteon, one single Haversian system in cross-section orientation. Periosteum, osteoprogenitor cells, osteoblasts inactive, osteocytes located in the bony lacuna. Here is active osteoblasts, periosteum, osteocytes, and probably somewhere here are also osteoclasts located in the house-shaped lacuna. Here the compact and trabecular bone, bony trabeculae. This is the bone marrow, the space which was filled with the red bone marrow and with the adipose tissue. Here is the compact bone, where the bony lamella are located in the parallel direction and calcified with hydroxyapatite calcium crystals. Trabecular bone with red bone marrow inside. Where the hematopoietic cells are developing and forming the precursor cells of the circulating blood. Metabolic role of the bone. Calcium ions, a key ion for all cells, is stored in bone when dietary calcium is adequate and mobilized from the bone when dietary calcium is deficient. Maintenance of proper blood calcium level involves activity of all three major bone cells, namely osteoblasts, osteocytes, and osteoclasts, and is largely regulated by subtle paracrine interaction among these and other cells of the bone tissue. Hormones affecting the calcium deposition and removal from the bone include parathyroid hormone, which indirectly stimulates osteoclasts and produced by the parathyroid gland. Elevated level of calcium in blood, and calcitonin hormone, which is produced by the parafollicular cells of the thyroid gland, which can inhibit osteoclast activity, lowering the blood calcium level. Osteogenesis means formation of the bone or osseous tissue It is also called ossification. There are designated two types of ossification, or osteogenesis: primary or intramembranous, and secondary or endochondral. Bones of the skull and jaws form initially by intramembranous ossification, with osteoblasts differentiating directly from progenitor cells in condensed membranes of mesenchyma. All other bones form from endochondral ossification, in which osteoprogenitor cells surround and then invade hyaline cartilage models of the skeletal elements in the embryo. This is the endochondral ossification. Diagram of primary or intramembranous ossification. Here you have the neurocranium, the mesenchyma, cluster of mesenchymal cells, from which the osteoblasts develop, producing the extracellular matrix, into which the osteoblasts incorporated are called osteocytes located in lacuna. And this is in the direct or intramembranous osteogenesis schematic view. Here you have different types of the cellular components of the osseous tissue: the osteoclasts, the osteoblasts, the active osteoblasts, inactive osteoblasts, osteocytes located in lacuna, bony lamella, mesenchyma, blood capillary with red blood cell, and endothelial cell forming the wall of the blood capillary, and cells of Marchand or pericytes forming the wall of the blood capillary. Primary intramembranous osteogenesis viewed by high magnification of light microscopy. Inactive osteoblasts, active osteoblasts, cuboidal in shape, forming bone tissue, synthesizing collagen fibers and concentrating the phosphate ions surrounding this ossification process. This is osteoclast, bigger cell, forming house-shaped lacuna, and osteocytes embedded in the hardened osteoid, hardened with calcium phosphate crystal. And here you have the mesenchymal cells, which will transform to either osteoblasts or later osteocytes. Intramembranous osteogenesis. Here you have the osteoclast, here you have osteocyte, here you have mesenchyma, here you have vessel of mesenchyma, here is mesenchyma, here is osteoid, red coloration, pink coloration, and osteoblasts located on the surface, secreting the extracellular matrix of the bone tissue, namely the enzymes which are responsible for the saturation of the bone tissue of osteoid with the calcium phosphate ions, calcium hydroxide, but calcium hydroxide with the crystals formation. Intramembranous primary osteogenesis on light microscopic view, the bony trabecula, the red bone marrow forming from the mesenchymal cells, and trabecule of bone, trabecular bone or spongy bone is very characteristic for this osteogenetic type. Hyaline secondary endochondral osteogenesis. First of all, the hyaline cartilage module as a template of future bone is created. Primary ossification centers in the aphysis of fetal long bones form when chondrocytes die after enclosure of the cartilage within the collar of woven bone, creating an initial cavity that is entered by the periosteal osteoblast and vasculature. So the bones that developing instead of the cartilaginous tissue, and the blood vessels are not bringing needed concentration of oxygen. Cartilage, the oxygen is deficient. When the oxygen is supplied by the blood vasculature, microvasculature bed, the chondrocytes, chondroblasts are dying, and instead of them from osteoprogenitor cells, the osteoblasts and osteocytes are developing. Later, secondary ossification centers develop similarly within the epiphysis, with cartilage of the epiphyseal growth plate between the primary and secondary ossification centers. The growth plates are the key to the bone elongation during childhood and are organized as an interrelated series of developing zones. Most distally is the resting, the reserve zone of typical hyaline cartilage. In an adjacent proliferative zone, chondrocytes undergo mitosis and appear stacked within elongated lacuna. The most major chondrocytes in these lacunas swell up, compress the matrix, and undergo apoptosis in the zone of hypertrophic cartilage closer to the primary ossification center. Space is created in the matrix, but these events characterize the zone of cartilage calcification when they are invaded by the osteoblasts, osteoclasts, and vasculature from the primary center. In the ossification zone, woven bone is laid down initially by osteoblasts and remodeled into lamellar bone. Summary of secondary endochondral osteogenesis: formation of hyaline cartilage template model, appearance of bony collar around the cartilage model, calcification of cartilage matrix, migration of periosteal cells into the cavity with growing blood vessels, structure of epiphyseal cartilage through the growth period, zone of reserve cartilage, zone of proliferation, zone of maturation, hypertrophy, zone of cartilage calcification, zone of ossification, cartilage resorption. Diagram of endochondral osteogenesis. First of all, formation of hyaline, fetal hyaline cartilage model. Then primary ossification center, forming the periosteal bone collar in the diaphyseal part of the future bone. Blood vessels of periosteal bud are entering the primary ossification center, and epiphyseal cartilages are site where the secondary ossification centers develop in epiphysis, developing compact bone, medullary cavities, and secondary center ossification where the blood vessels are delivering the oxygen and transforming the chondroblasts and chondrocytes to the osteoblasts and osteocytes. And final steps when the metaepiphyseal plate of growth, here you have its metaepiphyseal plate of growth, is disappearing, and the long bone stops increasing in the long length, and the regeneratory and physiological regeneration and degeneration due to the possible damaging of the tissues are developing through the whole lifespan. Rearrangement of the bone tissue, rearrangement of the primary woven bone to the lamellar bone, and formation of the osteon system is characteristic for the whole lifespan. Here, the cells and matrices of the primary ossification centers, osteoblasts, osteocytes, osteoids, and blood vessels. Endochondral osteogenesis in light microscopic magnification. Here you see the interface between the cartilage and developing bone tissue. Here you have its resting cartilage, proliferating cartilage, formation of the columns of the chondrocytes, the hypertrophic changes of the chondrocytes, the apoptotic changes of the chondrocytes and their transformation of the zones with the invading of the zones with the osteoprogenitor cells, osteoblasts, which are brought by the microcirculatory bed. And the calcification of the remaining chondrocytes, hypertrophic chondrocytes, and exchange them into osteoblasts and osteocytes. The secondary ossification center, the epiphyseal part of the long bone development, growth plate, meta-epiphyseal plate of growth, and the aphyseal ossification in between the secondary and primary ossification center is located the growing plate of the long bone. Growing plate of growth under the light microscopic magnification, the bone marrow located within the bone trabeculae, the secondary ossification center, blood vessels, and the intensively stained growth plate of growth of the bone. Endochondral osteogenesis epiphyseal plate of growth under the light microscopic magnification, the zone of resting cartilage is unchanged cartilaginous hyaline cartilage of auricular part of the epiphysis. Zone of proliferating cartilaginous cells, proliferating chondrocytes forming the columns of the chondrocyte. Zone of hypertrophic cartilaginous, hypertrophic chondrocytes increasing the volume. Zone of calcified cartilage, where the remnants of the cartilaginous tissue are becoming calcified. And zone of ossification, where the space filled primarily by the chondrocytes is being filled with the osteoblasts and osteocytes developing the lamellar, developing the bone and bone tissue. Epiphyseal plate. Epiphyseal plate of growth of bone. Growth of bones occurs throughout the whole life, with cells and matrix turning over continuously through activities of osteoblasts and osteoclasts. Lamella and osteons are temporary structures and are replaced and rebuilt continuously in the process of bone remodeling, by which bones change size and shape according to the changes in mechanical stresses which are attributed to the bone tissue. Bone repair after fracture or other injury involves the activation of periosteal fibroblasts to produce an initial soft fibrocartilage-like callus. The soft callus is gradually replaced by hard callus of woven bone that is soon remodeled to produce stronger lamellar bone. Here you have the diagram of woven bone transformation into lamellar bone. This is primary or initial bone formation within the lamella or primary membranous ossification. The cells are located in the neighboring cells are contacting one another with the processes. Successive. This is the early stages of the flat bone formation with the trabecula and periosteum of condensed mesenchyma, periosteum covering the condensed mesenchyma. This formation of the successive stages in the formation of secondary osteo and Haversian system during transformation of trabecular bone to compact bone. This is the subsequent stages of the formation of lamellae around the blood vessels and increasing in amount of the lamellae and decreasing the diameter of the central Haversian canal is leading to the formation of at least 6 concentric lamellae in large in thickness, characteristic for the formation of the osteons from the woven bone. Diagram of bone repair. The fracture hematoma forms. The damaged blood vessels are leaking the blood, which is covering the damaged space between the damaged parts of the bone. The fibrocartilage and soft callus formed instead of the blood cells. The formation of the chondroblasts and chondrocytes is leading to the formation of the fibrous cartilaginous callus with regenerative blood vessels supporting the osteoblast and oxygen supply, formation of the primary woven bone for the hard callus, and the transition of the woven bone to the lamellar bone with the formation of the compact bone at the break site, with callus resorbing during the time. Bone remodeling and repair. Woven bone, osteoblasts, osteoclasts, clearly visible giant cells of the resorption, characteristic for resorption, osteocytes located in the osteoid bone marrow, which is consisting of osteoprogenitor cells and developing to osteoblasts under the certain commands coming from interface, communication of the closely related cells, and then transformation of the woven bone to the lamellar bone, either spongy or compact bone consisting of the eversion systems. Joints are places where bones meet or articulate, allowing at least the potential for bending or movement in that portion of the skeleton. Joints with very limited or no movements are classified collectively as synarthroses, and freely mobile joints are called dysarthroses. Diarthroses have a joint cavity filled with lubricant synovial fluid enclosed with a tough fibrous articular capsule. End of the bones involved are covered with hyaline articular cartilage, which, as you probably remember, doesn't consist perichondrion, specialized connective tissue of the synovial membrane lines the capsule, which with folds extended into some areas of the joint cavity. Macrophage-like synovial cells remove wear and tear debris from synovial fluid. So instead of the epithelial cover, the macrophage, which are typical cells characteristic for the connective tissue, are covering the epiphyseal part of the joint bones. Fibroblasts-like synovial cells produce hyaluronate or hyaluronic acid that is moved in the synovial fluid with water from local capillaries, forming the synovial fluid that nourishes and lubricates the articular cartilage. Intervertebral discs allow limited vertebral mobility and consist of a large pad of fibrocartilage that cushions the adjacent vertebra. Each intervertebral disc consists mainly of the thick outer layer called fibrocartilage, forming a tough annulus fibrosus and a shock-absorbing water inner gel-like core, the nucleus pulposus. How it looks like is the anatomy of the synovial joint. These are epiphyseal parts of the contacting cells, contacting bones, the articular cartilage, which is hyaline cartilage, lacking the perichondrium, spongy bone, metaepiphyseal plate of growth, which is ossified, which is no more growing bone since the proliferation of the cells and the epiphyseal plate of growth is arrested. The synovial joint cavity and synovial membrane, which is providing the cavity with the synovial fluid. Here you have the specificity of the articular cartilage, where the calcified cartilage is within the depth. Then comes the collagen fibers in right angle to the surface of the arranged in the right angle to the surface of the top of the articular cartilage, and then near the top of the articular cartilage, the collagen fibers are changing their direction and becoming parallel arranged. And here in between these collagen fibers, there are ground substance and chondrocytes located. Complete absence of the perichondrium is characteristic for the hyaline cartilage forming the epiphyseal parts of the joints. And here you have the diagram of a synovial membrane histology. It's exclusion from the rule that instead of epithelial cells, macrophage-like cells are covering the surface of the epiphyseal part of the articular joint. Ground substance is represented by the macrophage and fibroblast-like cells. Fenestrated blood capillaries, which are producing watery fluid, filling the synovial fluid, synovial cavity, collagen debris, fibrils and debris, and fibroblasts located in the depth, within the depth of the synovial membrane. Here is the light micrographic representation of the synovial joint with synovial membrane, synovial cavity, with red bone medulla, in the epiphyseal part of the, this is capsula, this is trabecules, and cartilage, articular cartilage of the joint. Here you have joint capsule and synovium. Joint capsule, synovial membrane, synovial fluid, our articular cartilage, adipose tissue close to the spongy bone where the dead bone marrow was primarily existing. Articular cartilage under the light microscopic magnification. The surface of the articular cartilage, the bone tissue, and hyaline cartilage with chondrocytes located in the lacunas. So it is mostly the hyaline cartilage, but with exception of the structure, lacking perichondrium and having specific arrangement of the collagen fibers within this type of cartilage. And synovial membrane, showing you the joint cavity, the vascular elements of the synovial membrane, the macrophage arranged in the regular manner on the top of the surface of the synovial membrane, and connective tissue, collagen fibers, increasing the and forcing the joint capsule. This is the type of the joints, intervertebral joints, showing you the nucleus pulposus, annulus fibrosum, synovial joints in between the processes of the vertebra, and anterior and posterior longitudinal ligament, the dense regular connective tissue, keeping the whole vertebral column in needed condition, in the enforced condition. Again, the bone, two bones contacting the vertebral bones, contacting the intervertebral disc, annulus fibrosum, nucleus pulposum, cartilaginous tissue, auricular cartilage, and bony tissue with the spongy bone space, which is filled with the red bone marrow. This high magnification of the light micrographic representation of annulus fibrosum and nucleus pulposum of the intervertebral disc and the bone tissue at both sides of the cartilage of the intervertebral disc. Annulus fibrosum is, you probably remember that this is fibrocartilage. It's one of the two places in the human body where the fibrous cartilage is present. And here I give you the terminology which is concerning the pathology of the bone and cartilage, pathologies of bone and cartilage and tissues. And you can look in the internet, you can look in the textbook of pathological anatomy. This is the background showing you pathology of the particular details of the histology, normal histology, which were discussed right away on this lecture presentation. And the last slide is showing you the recommended reading for histology, including measure the easiest basic histology textbook for histology students, and the rest are also available in the Amazon.com. You can buy it, but the rest of all the editions are available and you can use them without charge. So this is all what I prepared to you for today's presentation. And this is the end of the lecture. Thank you for your attention.